Hello everyone and welcome to Let's Crack NEET UG English. The first thing first, we are going to check for the audio, and if audio is alright, then we'll continue with this. We are closing in for the completion of this chapter, that is photosynthesis. Today we will be talking about C4 cycle and the comparison part. We will also take up CAM, that is crassulacean acid metabolism, and then in short we will also talk about translocation of photosynthate. Photosynthate means the substance which is synthesized during photosynthesis. So is this audio clear? Just let me know, and if audio is okay, then we can get started. Okay, I have not heard from anybody so far. Okay, so I guess the audio is okay, and we are on this channel of Let's Crack NEET UG in English, and photosynthesis is the chapter that we are talking of. We have done in detail C3 cycle, C4 cycle, and why the C4 cycle is required in certain plants, and that is to avoid the loss of carbon dioxide due to photorespiration. And today's lecture is in continuation with that. Sorry. Let us go over this basic part. This is the schedule for this channel, and today is 21st. We are talking about this part, comparison of C3, C4 and CAM. So if you can look at this schedule, there are 2 more days in which we will be finishing this chapter, 22nd and 23rd. Sorry, there is some irritation in my throat. Okay, Reshma, good morning. How are you? Rahul is also there. Good morning, Rahul. Okay, so is the audio clear? I'm checking for the audio. Okay, Reshma, thanks for confirming. I was discussing the schedule with you for this channel, that is Let's Crack NEET UG English. We are on 21st, and 2 more days and we will be able to finish this chapter of photosynthesis. Now tomorrow's class we will be having this important topic for discussion, that is chemiosmosis and then the factors which affect photosynthesis. The last lecture which I have planned is for your doubt solving, and then we are going to have a quiz session so that we are able to assess whether everything is clear or not. Okay, so now Kalaya has joined. Hello everyone, and I have noticed that the time is 11 o'clock, but you join a minute or 2 late after that. Okay, let us come to the next sheet. This is my schedule for Let's Crack NEET UG. Only one lecture on this channel left, and after that I will be moving to the other channel. I'm already talking about human physiology on the channel Biology 101, and this chapter will be over by 30th of October. You are watching these videos of mine on various YouTube channels. I'm also taking classes on the app, that is the Unacademy learning app. And on this app, you will be able to get my special classes, and not only my special classes, but the teachers who are teaching physics and chemistry, their classes will also be available. Plus one more thing, the PDFs, like when we take a class and I draw a particular flow chart or a diagram for you, those PDFs are also available once you are on the app. So if you have not installed this app, please install it so that you are able to get all the benefits. And apart from daily live classes and tests and everything, the PDF is the benefit that I was talking about. See, this is what I was telling you from the very beginning, that this is the subscription plan and this is the pricing. But this is going to change from 24th, and yesterday also I showed you the new sheet. This will be applicable from 24th. So take advantage of the lower pricing. Right here for 12 months it is 27,000, and after 24th for 1 year it's going to be some 34,000 after the discount 10%, and you will get this discount only if you use the code. So do it as early as you can so that you get the benefit of all these things. Alright, Rahul says, ma'am, after 2 classes of heart chapter, will you move to other channel? Yes, I will. I have already started on the other channels like Biology 101, plus I have increased my hours on the special classes. But the special classes are only available on the app. So if you have not installed the app, you need to install the app. That is the first thing that you need to do. So how many of you have installed this app which I'm talking about? And how many of you have already taken the subscription plans? Because the things which like the special class and all those things where I'm taking genetics right now are available only on the app. Okay, you can message me whenever we get time in between, and we will start with our subtopic for today, that is comparison between C3 and C4 cycle. Now before we take the comparison part, did you revise C3 and C4 cycle, and do you have any doubt in these 2 cycles? So if there is a doubt, we'll discuss that first and then we will come to the comparison part. So send me your questions in case if you have. So I have not received any question. That means I think everything is clear, and while we are discussing the comparison between the 2 cycles, in case if you come across any difficulty or doubt, you may ask that. Now what we are doing is we are talking about certain points, and on those points we will be comparing these 2 cycles, and this is our simple table in which we compare it. So the first point is the site of C3 cycle in, let us write in C4 plants and in C4 plants. So what is the site of C3 cycle in C3 plants? where exactly the C3 cycle takes place. Now, if you start answering these questions, there will be two things. One, you will be able to revise it, and secondly, in case if you're not able to answer it, then that is going to be beneficial for you to discuss the doubts. So where do you think the site of C3 cycle in C3 plants? In C3 plants, C3 cycle takes place in mesophyll cells. And in C4 plants, C3 cycle takes place in bundle sheath cells. This is point 1. Rahul has answered mesophyll. See, when we are talking about this, your answering should be quick. Very good. Rahul has given C3 plants mesophyll cell and bundle sheath in C4. Perfect. Others have not answered. They need to be prompt and quick so that we know whether everything is clear to everyone or not. Now this is one. Now first, carbon dioxide acceptor. Okay, Reshma also says in C4 bundle sheath. Very good. Now answer this question. First, carbon dioxide acceptor in C3 plants. Which is that molecule that accepts carbon dioxide for the first time? C3 plants that we are talking of. And then we will take in C4. Rahul says RUBP. Very good. So in C3 plants, in mesophyll cells, ribulose biphosphate is the molecule which accepts carbon dioxide. And Reshma also gives the same answer. Now what about C4 plants? Which is that molecule that accepts carbon dioxide first? Is it RUBP or is it some other molecule? Okay, Rahul has given the answer, that is PEP, phosphoenol pyruvate. If you remember, phosphoenol pyruvate is a 3-carbon compound. It accepts carbon dioxide and then a 4-carbon compound is formed. So now let us come to our third point. Okay, the first stable compound, phosphoenol pyruvate. Very good. Rahul, Reshma have given. Mamta says add notes also. See, what kind of a note, what kind of notes are you looking at? See, what happens is whenever we teach, it is always beneficial that you jot down points while the teacher is teaching. And this is how you need to be preparing your notes. If you want the PDF, like this table which I am making, there are two options. Either you make the table with me while you're studying, or you just listen to it, and once the lecture is over, you want to use the or see the same table. So then the PDF of this will be available only on the app. And this is what I was telling you, that you need to install the app so that you are able to access these PDFs. I don't know how you make your notes. Normally what happens is when we make notes, the teacher is telling us something and then we write it in our own language. So that is how we do it. Okay, so first stable compound, Rahul and Kalai have given the answer as PGA, phosphoglyceric acid or phosphoglycerate in C3 plants. What about C4 plants? Which is that substance that is formed in C4 plants? And obviously that has to be a 4-carbon compound. So this is a 3-carbon compound, and in case of C4 plants, Reshma says 3-PGA, that's right, 3 is the position of the phosphate. Oxaloacetic acid, very good. So oxaloacetic acid is a 4-carbon compound, which is the first stable compound formed in C4 plants. Okay, Rahul has given the answer. Now let us take one more point. The next point, the 4th point, okay, Reshma has also given oxaloacetic acid. How many times is carboxylation taking place? Carboxylation. About carboxylation, if we have to talk of, in C3 cycle, how many times carboxylation takes place and where? Carboxylation is addition of carbon dioxide. So in C3 plants, how many times and where? See, this kind of thing helps us in revision also. So when we are answering these questions, all those steps in the cycle are also getting revised. Six times to form, oh, I'm not talking about the dark reaction or Kelvin cycle. I'm talking about carboxylation. So Rahul has given 6 times, that is Kelvin cycle. That is not the question. The question is, how many times is carbon dioxide getting added? Reshma also says 6 times in mesophil. Okay. Carboxylation, that is addition of carbon dioxide, takes place only once in mesophil cells, whereas in case of C4 plants it takes place 2 times, first in mesophil cells by PEP, and second time in bundle sheath cells by RUBP. So there are 2 places where carbon dioxide is getting added. Is it clear? Sunita has joined. Hello. Krish has also joined. Okay, so those who have joined little late, for them we are comparing C3 and C4 cycles on various points, and this is also helping us to revise everything. Mamta says, yes ma'am, this is for what? Okay, please write little larger. Okay, I'll write little bigger. So is the point clear? This is what we were talking about. We are talking of carboxylation, addition of carbon dioxide. So how many times it takes place, that is different. Like for synthesis of glucose, that is different. But we are talking of where is carboxylation taking place. When carbon dioxide is taken by RUBP, that is carboxylation. When it is taken by PEP, that is carboxylation. Okay? Okay, now let us talk about the enzymes. How many enzymes are required for this carboxylation? In case of C3 plant, there is only one, and that is rubisco. And in case of C4 plant, there are 2. One is PEP, carboxylase, and rubisco. Okay, so far all right. Now, is the font bigger enough for you to read? Who was the one who wanted me to write little bigger? So I think it is legible now. So we have written down the enzymes also. C3, location of C3. Now let us talk about how much of NADP or how many molecules of NADPH2 and ATPs are required for glucose synthesis. So requirement. How many ATPs? Mamta says, I'm from Nepal and I love the way you teach. Thank you, Mamta. I hope it helps. So are you in class 11th, 12th, or have you taken a drop? See, we have been together for many days now, and there are some students who are like almost every day in the class. So maybe what we'll do is you can just tell me about which class you are in, so that I would know that whether if you are in 11th class, this is for the first time you are studying. If you are in 12th, you have done this earlier. If you are a dropper, then that means you have done it more than 2 times. So that will be easier for me. Okay, so for C3 cycle, 12 NADPH2, and who has given the answer? Kalai has given the answer. Reshma has also given the answer. And 18 ATPs are required. And in C4 cycle, okay, Rahul has 12 NADPH and 18 ATP in C3 cycle. Sridhar has joined now. Good morning, Sridhar. Okay, I'm waiting for the answer for C4 plants. How many ATPs and how many NADPH2s are required? Okay, who has given the answer? Rahul, 12 NADPH2 and 30 ATPs. Reshma says 12 ATPs in C4, but what about NADPH2? Rahul, your answer is correct. It is 12 NADPH2 and 30 ATPs. So if you remember, in C4 cycle, there is extra 12 ATPs required. And that is how, because in C4 plant, C3 cycle anyways takes place. So C3, 18 ATP, and for extra C4, 12 ATPs more. And that is how we come to this number of 30 ATPs. See, all the points which I have written, most of you have answered these points. That means now you have understood the C3 cycle and C4 cycle. So this is how this comparison, or when we go over these things, they help in our revision also. Okay, Mamta says she is a dropper and you have been watching my lectures. Okay, that's good. So this means you have studied this C3, C4 cycle photosynthesis earlier also. Alright, so we have compared C3 and C4 plants, and now let us talk only about the examples. So can you give me the examples of C3 plants and C4 plants? Okay, who is giving me the examples of C3 plants and C4 plant? I would need at least 3 examples of each category. You can give me any one first. You can give me the example of C4 first or C3, but write down whether you're giving the example for C4 or C3 plants. I have given you the examples of C4 plants. Now I want to check whether you can give me the examples of C4 plants or not. Sorry, C3 plants or not. Or you want me to write down if you're not able to recall? I'm going to write down for C4. Maize, sugarcane, sorghum, chenopodium. These were some examples of C4 plants which I gave you. So now Rahul says sorghum, maize, portulaca, these are C4. Very good. Now what about C3? Sugarcane, amaranthus, millets, grasses. Perfect. Kalai has also given the answer. Reshma has also given the answer. Now can you give me 3 examples of C3 plants? And then we will come to the last point of comparison. Rice, wheat, barley, these are C3 plants. Sunflower, soya bean. See, if you exclude these, maize, sugarcane, sorghum, chenopodium, all other plants are C3 plants. So the plants which are visible to us commonly, like mango, neem, papaya, all these plants are C3 plants. But here we can write the examples as say wheat, mango, the common plants, apple, you can give any example except the C4 plants. Sunflower, soya bean, Abdul has given these examples. The last point which we are talking about is photorespiration. Now, whether photorespiration takes place in C3 plants and C4 plants or not, this is our answer. So is there photorespiration in C3 plants? And is there photorespiration in C4 plants? Do C3 plants undergo photorespiration? In C3 it takes place and absent in C4. Answer given by Rahul. Perfect answer. It takes place in C3 plants, and in C4 plants there is no photorespiration because they have the adaptation, they have Kranz anatomy and they have dimorphic chloroplast. Reshma has also given the answer, Priya has also given that answer. No photorespiration in C4 plants. So now when we are comparing these two cycles, or these two plants rather, C3 plants and C4 plants, we have actually revised the complete C3 cycle and C4 cycle. Okay, so this is the difference what we talk of. Now if you have read NCERT, in NCERT also there is a tabular thing which is given, and where we are asked to fill the blanks on the basis of the plants, and it is made, they have made it very easy for us. Like they have given the options from where we have to choose from. So what we'll do is we'll fill few boxes and the other boxes will be homework for you. I'm going to do couple of boxes with you. Okay, say the first one. Okay, let us start with the first one. What we did was our way of comparing C3 plants and C4 plants. Here we will do what is given in NCERT. Cell type in which Kelvin cycle takes place, and we have to choose from mesophyll and bundle sheath. So in case of C3 it is mesophyll and here it is bundle sheath, right? Okay, then let us come to this, which is the primary carbon dioxide acceptor. In case of C3 it is RuBP, and in case of C4 it is phosphoenol pyruvate. Number of carbons in the primary carbon dioxide acceptor. In case of C3 plant, which is the primary carbon dioxide acceptor, RUBP, and RUBP is a 5 carbon compound. And in case of C4 plants, the primary carbon dioxide acceptor is phosphoenol pyruvate. It is a 3 carbon compound. Which is the primary carbon dioxide fixation product. See, we have done the same points in comparison, but when you go through the bookish language is slightly different. So here also our answer is going to be pretty much same, but looking at the bookish language, we have to reach to that answer. So which is the primary carbon dioxide fixation product? In case of C3 plants, it is phosphoglyceric acid, and in case of C4 plant, it is oxaloacetic acid. Alright, now let us take this point. Does the plant have rubisco? C3 plants, do they have rubisco? The answer is yes. Do C4 plants have rubisco? Answer is yes. Because C3 cycle takes place in both, and for C3 cycle to take place, rubisco must be present. Now, does the plant have PEP, that is PEPCO here they are talking of, phosphoenol carboxylase. So, do C3 plants have PEPCO? Actually, not always. They don't need it because it is rubisco, and when rubisco is not working properly, there is photorespiration and there is loss of carbon dioxide. But here it is yes, and here it is not always. Okay, so this is what is given, the tabular thing, the comparison part in NCERT. This is the half part of it, and we have discussed few points. The other points which I have left blank, those you have to fill in on your own. Now this is the continuation of the table. Which cells? This is for C3 plant and this column is for C4 plants. Which cells in the plants have rubisco? In case of, and we have to choose from here. So in C3, these are mesophyll cells, and in C4, rubisco is present in bundle sheath cells. Okay. Now, whether photorespiration is present at high light intensities, whether photorespiration is present or not. Photorespiration is seen in case of C3 plants, and that is when light intensity is high. What happens when light intensity is high? High light intensity. If light intensity is high, which is a stressful condition for the plant, stomata close. And when stomata close, the oxygen molecule which is given out during light reaction remains trapped inside. And if it is trapped inside, that means oxygen concentration is going to increase. And we know when oxygen increases, rubisco's behavior changes. High light intensity indirectly results in increase in temperature. So at high light intensity, photorespiration is definitely going to increase. So here it is high. And what should be the answer in case of C4 plants? It is negligible. almost not there. So, and examples. So, examples of C3 and C4 also we have written. So, this is what you have to fill in the remaining boxes. Okay, so we have seen the comparison of C3, C4 plants in our own language, and then when we do it in a bookish language, comparison is same, but the points, they are written in a slightly different manner. So this helps us in revising C3 and C4 cycles. Now let us come to another pathway which is called CAM. CAM basically stands for Crassulacetic acid metabolism. And this is a slightly different pathway which is seen in succulents like opuntia. It is also seen in pineapple, and it is also seen in the members of Crassulaceae family. And that is how the name Crassulacetic acid metabolism. Now what happens in these plants is they have changed the timing of opening and closing of stomata. Crassulacetic acid metabolism takes place in such plants which have scotoactive stomata. What are scotoactive stomata? There are messages, somebody wants me to take questions in between, and somebody wants me not to take questions in between. See, what happens is, if you have to just think from the perspective that if it was a live class where you were sitting in front of me, then what happens is a teacher is able to assess whether the students are understanding or not from your facial expressions. So when I look at my students who are sitting in the class, just by looking at their face, I'm able to assess that something is not clear, the person is not able to understand. But in such type of a class where I'm not able to see you, what is that means or method by which I would be able to assess whether you are able to understand it or not? That is the purpose of a live class, so that as soon as the doubt arises, I should be able to answer that question. Otherwise, the doubts will keep accumulating, and at the end of the chapter, you would still have those 5-10 points which are not clear. And that is why I need to ask you in between the lecture whether you have understood this thing or not. And Mamta, have you attended earlier lectures, earlier chapters which I have done on other channels or not? Because in between I keep giving questions, there is homework which is given, and by the end of the chapter there is always some multiple choice questions which are given. And today also when I shared the schedule, this is what I told everybody, that once we finish photosynthesis, we are going to have a quiz. And obviously we take multiple choice questions. Okay, so the purpose of asking questions in between is whether everything to everyone is clear or not. Okay, so we were talking about scotoactive stomata. Now, let me ask you, do you know what exactly we mean by scotoactive stomata, and how many types of stomata have you studied? There are basically two types of stomata. One is photoactive and other is scotoactive stomata. Scotoactive stomata are the ones which open during night and remain closed during day. Now why? Okay, I have given you the answer, Rahul. If you were not aware of this, now you know what is meant by scotoactive stomata. Reshma knew it, opening during night. Okay, so scotoactive stomata are the ones which open during night. Normally, plants have stomata which are photoactive. They open during day and remain closed during night. But we are talking about succulent plants. These plants, they grow in desert areas, extremely hot conditions. And if stomata are open during daytime, then what are the problems these plants are going to encounter? See, these plants are growing in dry, arid areas. Water is anyways a problem there. So there is very less water available to them, and secondly the temperature is high. So whatever that little water is, whatever water they absorb will be lost through transpiration. So what option these plants have? The option is that they change the timing of opening and closing of stomata so that they are able to avoid this loss of water through transpiration. Now, how is this crassulacean acid metabolism going to take place? We will draw two diagrams, one which is showing that what happens during daytime, and second is what happens during night time. So here we are talking about first diagram, which is what happens during night. So here is this stomata that I have drawn. This is during night, and because it is having scotoactive stomata, the stomata are open. Stomata open. So if stomata are open, then only gaseous exchange is possible. And this is the mesophyll cell. In mesophyll cells, there are vacuoles, chloroplast, all those structures are there. So now what happens during night time? During night time, carbon dioxide is taken in because stomata are open. This carbon dioxide reacts with water in presence of carbonic anhydrase to form carbonic acid. This carbonic acid dissociates into H+ and bicarbonate ions. This part we talked about when we were talking of C4 cycle. This bicarbonate ion is accepted by phosphoenol pyruvate, which is a 3-carbon compound, and oxaloacetic acid is synthesized. A C4 compound is synthesized. So now this oxaloacetic acid is formed. This oxaloacetic acid is converted into malic acid. And this structure which I have drawn here is a vacuole. And all throughout the night, the same process is going to take place, and this malic acid is stored inside the vacuole. So now for the entire night, carbon dioxide is taken in. It is first converted into oxaloacetic acid, then into malic acid, and then stored in the vacuole. So during night time, carbon dioxide is stored. How is it stored? Stepwise manner. First it reacts with water to form carbonic acid. Here the enzyme which is helping is carbonic anhydrase. and then malic acid is stored in a vacuole. This is what is happening during night time. Now let us see what happens during daytime. Now during day the stomata is closed. During daytime, here stomata closed. So because stomata is closed, there is no gaseous exchange. Here we made this vacuole in which all throughout the night malic acid was stored. So now during daytime, there is no entry of any gas. There is no carbon dioxide coming in, but there is malic acid which is accumulated in the vacuole. Now this malic acid comes out of the vacuole. It dissociates into two parts. It's a 4-carbon compound. It dissociates into carbon dioxide, 1 carbon is released, and pyruvate, which is a 3-carbon compound. This carbon dioxide is used in C3 cycle to fix that carbon dioxide, and glucose will be synthesized. That means C3 cycle will take place, and this is daytime, so light reaction is also taking place. So as soon as light reaction takes place, ATP, NADP, H2 are synthesized and carbon dioxide is released inside the cell. Carbon dioxide is accepted by that RUBP. Rubisco is already there. C3 cycle will take place and glucose will be synthesized. Now this pyruvic acid has to be converted into phosphoenol pyruvate, because during night again this phosphoenol pyruvate should be available to accept more and more carbon dioxide, and we know for this conversion we would need 1 ATP which will break down into 1 AMP, and this is the same thing as 2 ATPs. So how many ATPs and NADPH2s will be required for this? See, if it is C3 cycle, then 12 NADPH2 will be required in any case. 18 ATPs will be required in any case in C3 cycle. This is fixed. And to regenerate PEP from pyruvate, if you have to regenerate one molecule, 2 ATPs will be required, and to synthesize one glucose we have to do it 6 times. So plus 12 ATPs. So total will be 12 NADPH2 and 30 ATPs. So this is the requirement is very much similar to what happens in C4 plants or C4 cycle. Only difference is in CAM, that is crassulacean acid metabolism, the plants have an adaptation that they have scotoactive stomata. So they have avoided the loss of water, because for them the most important factor is water. Because if there is no water, no photosynthesis, because water is the raw material for photosynthesis. Because hydrogen, which is used to make that reducing power, comes from water, and oxygen is given out. So this is CAM or crassulacean acid metabolism. Now tell me whether this pathway is clear or not. Is this pathway clear? And how is it different from C4? Everything is taking place in the mesophyll cell. So the cells that we have drawn are mesophyll cells. Okay, Rahul says clear. What about others? And we have also written the examples like succulent plants like opuntia or cactus, pineapple, members of Crassulaceae family, and some members of Euphorbiaceae family also show this. What about others? Is this CAM pathway clear? Is there any doubt in this particular pathway? Priya also says clear. So now C3, C4, CAM, photorespiration, all these pathways are done. And now we can easily understand what is the difference between all those things. So 3 people or 3 students have given the answer that it is clear to them, Crassulacean acid metabolism. That is Rahul, Priya and Reshma. I don't know about others, like Kalai has not answered, Mamta has not answered. So Abdul has not answered, so you need to tell me whether it is clear or not, so that we can move to the next part. Okay, so most of you are saying it is clear. Let us now talk about the next part, translocation of photosynthate. What exactly is our photosynthate? Photosynthate is the substance or organic matter which is synthesized during photosynthesis. So product of photosynthesis. Now here there is something very, very important. What is synthesized is glucose. Glucose is synthesized during photosynthesis. Glucose is a highly reactive molecule. It's a reducing sugar. It is translocated. Translocated means from the place where it is synthesized to the place where it is to be stored. That process is translocation in the form of sucrose. Sucrose is a disaccharide and is non-reducing sugar. So this is a reducing sugar and so is reactive. Sucrose is the disaccharide which is translocated and it is a non-reducing sugar. And that is why carbohydrate which is synthesized as glucose is translocated as sucrose. And then this sucrose is converted into starch and is stored. So this is stored. It is a polysaccharide. And it is always beneficial to store carbohydrates in the form of polysaccharides. Okay, Kalai says he had some network problem and will watch and reply. Okay, so if it is clear, just message that it is clear to you, and if you have any doubt, then just message that doubt. And Mamta is saying how many lectures are left? Mamta, did you attend this class from the very beginning? Because in beginning of every lecture, I give you the schedule of the next 4, 5, 6 days. After this lecture, we have 2 more lectures for photosynthesis, and then we will start with another chapter. Okay, so what is meant by this translocation? Translocation is movement from one place to another. What is that one place? From place of synthesis to the place of storage. So where is the carbohydrate synthesized? That is leaf. And where is it stored? Depending upon the plant. It can be stored in leaf, it can be stored in fruit, it can be stored in stem, and it can be stored in root. I'm sure you have studied morphology of flowering plants also. Okay, I'll take your question, Rahul, in a minute. What exactly happens here? So we are talking about place of synthesis, that is from leaf. When it is sent to the place of storage, that process is called translocation. And depending upon the plant, it can be stored anywhere. Like in which plant do you think that carbohydrate or the food which is synthesized is stored? In the leaf. Cabbage. And that is why we eat the leafy part of cabbage. Fruit, most common, any fruit, mango, apple, etc., any fruit. Stem, where in stem is carbohydrate stored? Sugarcane. And that is why we take or consume the food stored in the stem of sugarcane. Where is it stored in the root? Any example? Carrot. So it is very simple. If you are eating leafy part of a particular vegetable plant, that means the food is, why do we eat that vegetable or that particular part of the vegetable? Because we are getting the food or nutrition from that plant. So we consume leafy part of cabbage because it is the leaf where the food is stored. We eat potatoes. What is that potato? It is stem, modified stem. So why are we eating potato, that tuber, and not the leaf of potato plant? Because the food is stored in that tuber, that is in the stem. So in sugarcane we can also write the example of potatoes. Why are we eating the root of a particular vegetable and not the leaf or not the stem or any other part? Because it totally depends on which is that part where the food is stored. And we eat those vegetables because we want the food. Rahul has given potato answer, carrot, radish. Reshma has given sweet potato. Is it a stem or a root? Reshma, answer this question. You have given the answer as sweet potato. Is it a stem or a root? Where should I write it? Should I write it in front of stem or should I write it in front of root? Okay, I want to know anybody else who wants to give the answer. They can also give the answer. Sweet potato, is it a stem or a root? This is from morphology. Reshma says root. Very good. So potato is stem and sweet potato is a root, a modified root. Okay. Afreen says stem. No, potato is stem because characteristic feature of stem is that it has nodes and internodes, and sweet potato is a modified adventitious root. We will be talking about all these things when we come to morphology of flowering plants, when we take that chapter. Okay, now coming to the question which Rahul asked. What is the mechanism glucose is getting changed into sucrose and then into starch? See, what happens is glucose is a reducing sugar. It is highly reactive, and if you translocate that reactive molecule, it is going to react with the molecules or the structures which are in its path. So you have to change it into something which is more stable. And when sucrose is formed, sucrose is a disaccharide. So disaccharide sucrose is formed by combination of one glucose and one fructose molecule by formation of a glycosidic bond, and that results in formation of sucrose. And sucrose is non-reducing because the bond which is formed is between the keto group of fructose and aldehyde group of glucose. Keto group and aldehyde group are the reducing groups, and because these two groups react to form the glycosidic bonds, both the functional groups are gone, and that is why sucrose is non-reducing. And this sucrose synthesis by formation of glycosidic bond is by dehydration, that is, there is loss of one water molecule. That means 2 atoms of hydrogen, 1 atom of oxygen less. And when sucrose is converted into starch, say there are 1000 sucrose molecules or glucose molecules for that matter. So suppose there are 1000 glucose molecules which have combined by glycosidic bonds to form starch. Then in that case 999 water molecules are given out. That means 999 oxygen atoms and double of that hydrogen atoms. So the polysaccharide like starch is a compact molecule, and it is always easier to store a compact molecule. Okay, so I hope your question is answered and it is clear to you. Afreen is asking, explain again. What is that thing which you want me to explain, Afreen? And Rahul, is the explanation clear to you, the question which you asked about polysaccharide, disaccharides and all those things? Very good, it's clear. Okay, so this is what we had planned for today. CAM, difference between C3, C4. Don't forget to complete that table from NCERT and also read the entire chapter from NCERT, because in a day's time we will be starting with doubt solving. We have one more lecture where we will be talking about chemiosmosis, that is tomorrow, and day after doubt solving, and then we will have a quiz. So prepare this chapter, go through all the PDFs, those are available on the app. If you have not installed the app, please do that, and don't forget to subscribe as early as you can, because the plans are changing from 24th, and use this code so that you are able to get that 10% discount. Okay, others have also responded that it's clear. Kalai is asking, ma'am, in Biology 101 you will take in English. It is 80% English, but like the filling things are in Hindi, just the normal conversational things, but terminology is all going to be English. You can attend couple of classes, you will understand it. Okay, so I'll see you tomorrow, same time, and we'll continue with this. And don't forget to complete that homework from NCERT and revise this chapter, because we are going to have a quiz day after tomorrow. All right, so till that time, till tomorrow, take care. Okay, bye bye.